now that we've windowed our embryo and seen the beating heart, it's time to sacrifice this embryo. Remember, if I put this back in the incubator, it wasn't going to survive anyways. So to sacrifice this embryo, what I'm going to do is I'm going to immerse it in paraformaldehyde. Paraformaldehyde is a fixative. I'm using 4% paraformaldehyde. And what that does is it acts like a glue that holds all the RNAs, all the proteins, all the cell membranes exactly in place where they are right now. So I'm going to use this small vial of paraformaldehyde to insert the embryo. So how am I going to do that? Well, first of all, I want to get the embryo out of here, and I'm going to place the embryo into something called chick wringer solution. This is basically just a salt solution. Like when the nurse gives you an IV, you've got a saline solution going in because we want something that has the same concentration as what is in the chick cells. So the first way I'm going to try to do this with is with as little mess as possible. If I have to crack the egg, I will. But right now I'd like to try to just lift the embryo up out by sticking forceps down in. And I'm going to cross my forceps and make a little cradle for the embryo. And pick the embryo up and place it into this chick wringer solution to remove the yolk from it. There's my embryo. Can you see the little question mark, the little letter P? The head is to this side, the heart is right here, and the rest of the body goes down this direction. Now I could have just cracked the egg. It would have made more of a mess though. Okay, so I'm sort of lucky that I was able to get this. Okay, it wasn't luck, I've practiced. But now what I want to do is I want to make sure that this embryo is ready for the analysis that we would do as a developmental biologist. For example, we might want to find out what genes are being expressed. Differential gene expression is the most important part of developmental biology. It explains how every cell is different from another. So what I want to do is I'm going to put this under the microscope, and what I'd like to do is make sure that all the yolk proteins have been pulled off of this embryo. The embryo also comes with a chorion. This is like a straitjacket that protects the embryo while it's in the egg. So I want to find that chorion and make sure that I can pull it off so that we'll have pure embryo rather than part of the egg or the yolk. And I'm going to turn another light source on here. There, that might help me find it. Yes, I am right above the embryo, and now I've got it in focus. Why don't you zoom in here and then try to get it through the microscope as well. There we go. That is our embryo, head at the top. Yep. And then the body curves off to the bottom right. Yep, just like that. So notice that all vertebrate embryos, whether mammals or birds like this chicken or snakes or any other reptile or amphibian, they all develop their brain first. So at these early embryonic stages, this embryo is mostly head and only has a little bit of the rest of the body. So what I'm going to do here is I'm going to find a small piece of tissue that is hanging off of the brain, and I'm going to pull this coat off of him. Remember, I said it's a little bit like a straitjacket. So I'm going to grab this tissue with these fine forceps, and I'm just going to disrobe him. Make sure that the embryo is cleared of any connection to yolk. Okay, well done. See if you can get a close-up again through here while I hold the embryo in place. I want to see if you can see its eyeball. The eyeball has started to develop. I'm holding the middle of the embryo about where its heart is, and just above my forceps, There's a little dark circle, and that's its eyeball. Final step is to transfer this cleaned embryo into the fixative. The embryo is now floating around in the fixative. It's right there. This embryo contains all the RNA, all the DNA, all the proteins, all the membrane phospholipids that we might want to study at a molecular level. So we can extract this from the embryo, or we could put this embryo onto a microscope slide. And you, developmental biology class, have already looked at whole mounts of chick embryos. That would be just like if I put this embryo right here onto a microscope slide and stained it so that it was colored, and we put it, so every cell was either pink or the nucleus was dark, and we could put them under a microscope and look at them.